We now turn to the physiology of hearing in Chapter 10. Much like the anatomy, the physiology is very similar across different species of animals. The ear acts as a transducer. It takes pressure changes in the air, which is what sound is physically, and turns it into a neurological signal that's electrochemical in the body. And that's the idea of a transducer, converts one type of energy into another. In terms of the mechanism behind our hearing, we process the acoustic signals of speech over quite a wide range of characteristics. So our hearing can cover quite a large range of sound pressures, which we measure for practical purposes using a dB scale that covers a sound pressure range from 20 to 2,500,000 micropascals, so a factor of more than 100,000 from the lowest to the highest pressure. We can also hear a frequency range spanning from 20 to 20,000 hertz, where that's a unit for sound frequency. That covers a 10-octave range of sound frequencies, where each octave is a doubling of frequency. So once again, we have quite a large span that our auditory system can process. The way we process sound, it turns it into a neurological representation that is called tonotopic, meaning similar frequencies get processed in nearby locations in the neurological system. And it also encodes temporal information, the fact that the acoustic signal changes over time as we produce speech or as we're listening to music or any of the other sounds we might be interested in. Audiologists use a variety of instrumentation to assess parts of the hearing system. So tympanometry uses air pressure to assess how the middle ear is working by applying air pressure to the tympanic membrane. An audiometer provides sounds at specific frequencies and intensities, which people respond to, indicating whether they can hear them or not. The ear itself actually produces noise in response to sounds being entered into it. These are called otoacoustic emissions, and we can use these to assess ear function. This is what's done in screening babies to see if they may have a hearing loss before they could provide any feedback overtly about whether they hear something or not. And then once we get into the neurological processing of sound, we have auditory brainstem responses, which are neurological signals that we can measure through the skin, reflecting activity in the brainstem involved with hearing. And then over a somewhat longer time window, up to a few hundred milliseconds, we have event-related potentials or evoked potentials, where again we can measure electrical activity in the cortex through the skin as sound gets processed in the brain. The function of the outer ear in the auditory system is primarily to collect sound. So we have the pinna out there that acts a little bit like a funnel to bring acoustical information into the external auditory meatus, that auditory tube. That funnels sound down to the tympanic membrane. And sound is basically variations in air pressure. So those variations in air pressure then push on the tympanic membrane. The outer ear acts as both a resonator and a filter. It enhances sounds between about 1,500 and 8,000 hertz, with peak enhancement around 2,500 hertz due to its depth, basically, the fact that it's about 2.5 centimeters deep on average. The outer ear also reduces the amplitude of sound that comes from behind the head, so it funnels sound in more from in front of the head, which gives us a little bit of sound orientation and focus ability. The middle ear is an important transmission chain for that acoustic energy, that sound pressure from the air in the outside world to the fluid inside the cochlea. Part of what it does is overcome impedance, impedance being the resistance to the flow of energy. The way it does this is by increasing the pressure that arrives at the cochlea compared to the pressure that's at the tympanic membrane. We can understand a little bit how it does this by some basic physics. Pressure is a measure of the amount of force applied to an area. So we can increase pressure either by increasing the amount of force that's there or decreasing the area that that's applied to. And both things happen in the middle ear. So there's a little bit of a whip crack type effect of the movement of the tympanic membrane affecting the manubrium that increases the amount of force applied to the manubrium. When we look at the ossicles, we have a slightly larger piece on the malleus and the manubrium, which eventually will help move the incus and the long process, which applies to the stapes. And the fact that the moving part of the malleus is a little bit longer than the moving part of the incus increases the force that's applied there as a lever. The tympanic membrane is 17 times bigger than the oval window, so taking the pressure applied to the tympanic membrane and focusing it down onto a much smaller area for the oval window will increase the pressure. Overall, we get a gain of about 30 dB in the common intensity scale we use for looking at sound, which is about a 600 times increase in the pressure that's applied on the tympanic membrane versus what's applied to the oval window. And that's useful because the fluid that's inside the cochlea is harder to move than the air from the outside world. So we need to amplify that pressure to get an equivalent amount of fluid movement. When we look at the inner ear, we have both our acoustic analysis of what's going on in the sounds that we hear, as well as the vestibular system, which tells us things about how our head is positioned and how we're moving. So when we go to look at what the inner ear does, there are quite a few components actually between the vestibular mechanism, between what happens physically as part of the auditory mechanism, and then what happens neurologically as we move into the auditory neurological pathway. The vestibular mechanism gives us a lot of information that's part of our proprioceptive sensory system, which is how we understand where our body is positioned in space. The anterior or superior semicircular canal can sense a nodding type movement of the head, so up, down, more or less. The lateral semicircular canal senses the head shaking in like a no type movement, left to right. The posterior semicircular canal gives us the third dimension, best described as sensing a head tilt from left to right. So we take this information about head position, we put it together with sensory feedback we get from our joints, knowing what the position of our joints are, and from our muscles, knowing how contracted our muscles are, along with what we can see to give ourselves an overall mental picture of the layout of our body in space. The way the vestibular mechanism senses these positions and movements is on the basis of fluid movement that happens within the system. So we have stereocilia inside the ampullae in each of the semicircular canals that we can translate to movement or change in posture of the head in three-dimensional space. In addition, we've got the utricle and the saccule that help us sense movement and change in position. Again, we have stereocilia in each of the maculae that sense this fluid movement, and these can sense acceleration and deceleration in the movement of the head or the movement of the body. The utricle being more sensitive for horizontal movement and the saccule being more sensitive to vertical movement. So between the three-dimensional position of the head in space and sensing changes in position forward and backward or upward and downward, we can get a pretty good idea of how our body is positioned or moving. In the auditory part of the inner ear, we have the cochlea breaking the speech down into spectral frequency-based and temporal time-based components. We get this information from incoming sound waves by those sound waves being translated into fluid waves, and those fluid waves affecting the basilar membrane. The fluid wave would start at the base of the cochlea, which is near the oval window, and then travel down the spiral of the cochlea until it reaches the apex. Depending on the frequency of sound, there is going to be an area on the cochlea that gets maximally displaced by that sound, basically on the basis of the fact that the cochlea gets thicker as you go from the base toward the apex. So different thicknesses of the cochlea are going to get affected by different sound frequencies differentially. These mechanical events get converted into neurological events by the hair cells being excited. So as that fluid wave moves the basilar membrane, the stereocilia on the hair cells, which are connected to the tectorial membrane and to one another, are going to get bent. And when they get bent, that causes chemicals in the cell to be released, which will trigger a neurological signal. So to continue understanding what happens in the physiology of hearing, we'll have to shift over to neurological events.